Welcome to this lecture on animal biotechnology. Today, we're going to explore one of the most fascinating and powerful tools in modern science, restriction enzymes. These are often called the molecular scissors of biotechnology. Without them, the entire field of genetic engineering, from creating life-saving medicines to developing genetically modified organisms, would simply not exist. Let us dive in and understand how they work. To understand restriction enzymes, imagine you have a massive, miles-long ribbon with millions of letters printed on it, representing a complex genetic code. Now, suppose you want to cut out one specific sentence from this massive ribbon to paste it somewhere else. You cannot just rip it randomly. You need a highly specialized pair of scissors that only cuts exactly when it reads a specific word, like the word apple. In the world of animal biotechnology, we have exactly this kind of tool, and we call them restriction enzymes. These microscopic molecular scissors patrol the DNA strands and make precise cuts only at very specific sequences. Now that we understand the basic analogy, let us look at where these tools actually come from. Formally, restriction enzymes are known as restriction endonucleases. They are specialized proteins naturally produced by bacteria. But why do bacteria make them? Bacteria originally evolved these enzymes as a brilliant defense mechanism to protect themselves. When a virus attacks a bacterium and injects its viral DNA, the restriction enzymes act like a security force, chopping up and destroying the invading viral DNA before it can take over. Scientists have successfully hijacked this natural bacterial defense system, repurposing it to cut DNA exactly where we want it for genetic engineering. So, how exactly do these enzymes make their cuts? Restriction enzymes do not just cut anywhere. They cleave DNA molecules at specific, highly defined sequences called recognition sites. Interestingly, these sites are usually palindromic, meaning they read the same forwards on one strand as they do backwards on the complementary strand. When the enzyme finds its specific recognition site, it binds to the DNA and breaks the strong phosphodiester bonds that hold the DNA backbone together. Depending on the specific enzyme used, this cut can leave sticky ends with overhanging single-stranded DNA or blunt ends where the DNA is cut straight across. Understanding how they cut leads us to why this is so revolutionary. The discovery of restriction enzymes made recombinant DNA technology a reality. Because these enzymes cut DNA at predictable and exact locations, scientists can use them to cut a valuable gene out of one organism and paste it into another. For example, the human gene for insulin is cut out using restriction enzymes and inserted into bacterial DNA, allowing bacteria to produce life-saving insulin for diabetic patients. If we did not have restriction enzymes, we would not be able to create genetically modified animals, develop gene therapies, or even diagnose many genetic diseases. While biology might seem free of math, we actually use probability to predict how often a restriction enzyme will cut a piece of DNA. This brings us to the formula for expected fragment length. L equals 4 to the power of n. Conceptually, this formula calculates the average distance between cuts by looking at the probability of a specific sequence appearing by chance. In this equation, L represents the expected average length of the DNA fragments after cutting, measured in base pairs. The variable n represents the number of base pairs in the specific recognition sequence of the restriction enzyme. The number 4 comes from the 4 possible nucleotide bases: adenine, thymine, cytosine, and guanine. If the value of n increases, meaning a longer recognition sequence, the expected fragment length L increases exponentially because the sequence becomes much rarer. Now that we have our formula, let us see this with a concrete example. Suppose you are working in a genetic engineering lab and need to digest a long strand of unknown DNA using a restriction enzyme called SAU3AI. We are given that this is a 4-cutter enzyme because it recognizes a specific 4-base pair sequence, GATC. We are asked to find how often, on average, this enzyme will cut the DNA. We choose our expected fragment length formula because it models the statistical probability of finding that exact 4-letter combination. We substitute the value n equals 4 into our equation. We are raising the number 4, representing the 4 possible DNA bases, to the power of 4. Calculating this gives us a final answer of 256 base pairs. Physically, this means the enzyme chops the DNA into very small, frequent pieces, which is highly useful for building a comprehensive genomic library. Let us advance to a more complex experiment using the famous restriction enzyme EcoR1. We are given that EcoR1 has a 6-base pair recognition sequence, GAATTC, making it a 6-cutter. We need to find its average fragment length to establish a baseline frequency for its specific target. Again, we use our probability formula. We set the variable n equals 6 because the enzyme looks for a sequence that is exactly 6 letters long. Raising the base 4 to the 6th power gives us the average distance between these specific 6-letter words in a random DNA sequence. The final answer is 4,096 base pairs. Notice how requiring just 2 extra specific bases makes the sequence much rarer in the overall genetic code, resulting in DNA fragments that are significantly longer and fewer in number compared to the 4-cutter. Now that we have calculated the average fragment length for EcoR1, we can take the next step in planning our experiment. Once we know how often an enzyme cuts on average, we can use that information to find the total number of pieces it will produce from a specific DNA molecule. This 2-step approach is standard practice for molecular biologists planning a DNA digestion experiment. To do this, we must know the total size of the genome we are cutting. This calculation is absolutely critical for scientists when planning experiments like gel electrophoresis because it helps them predict exactly how many bands of DNA they should see glowing on their gel, confirming if the experiment worked correctly. This brings us to the formula for calculating the total number of fragments. Conceptually, this is a simple division problem. We want to know how many times our average fragment length fits into the entire genome. In this formula, F represents the expected number of DNA fragments. The numerator is T, which is the total length of the DNA strand we are cutting, measured in base pairs. The denominator is L, the expected average fragment length we just learned how to calculate, also in base pairs. If the total genome size increases, the number of fragments F will increase. Conversely, if we use an enzyme with a larger average fragment length L, the total number of fragments F will decrease, giving us fewer, larger pieces of DNA. Let us apply this formula to our EcoRI experiment. We are given a viral genome that is known to be exactly 49,152 base pairs long. We are asked to calculate exactly how many individual DNA fragments we expect to get after the EcoRI enzyme finishes cutting the entire genome. We choose our division formula because it tells us how many times the average cut distance fits into the total length. We take the total genome size of 49,152 and place it in the numerator. We then divide it by our previously calculated average fragment length of 4,096 in the denominator. The final answer is exactly 12 fragments. Physically, this means if you digest this specific viral genome with EcoRI, you can expect the DNA to be chopped into roughly a dozen distinct pieces. Now that we have done the math, let us visualize exactly what EcoRI is doing at the molecular level. In this diagram, you are looking at a double-stranded DNA molecule. Notice the top strand, reading from 5' to 3', contains the specific sequence GAATTC. The bottom, complementary strand, reads the exact same sequence in reverse, making it a palindrome. The arrows indicate the exact cleavage points where the EcoRI enzyme breaks the phosphodiester bonds, specifically between the guanine and adenine bases on both strands. On the right side of the diagram, you can see the result of this staggered cut. The DNA has been split into 2 fragments, leaving overhanging unpaired bases reading AATT. These are the sticky ends we discussed earlier, which are crucial because they allow scientists to glue this fragment to another piece of DNA with matching sticky ends. We have covered a lot of ground today, so let us review our key takeaways. First, restriction enzymes are the indispensable molecular scissors of biotechnology, allowing us to cut DNA at highly specific, predictable locations. Second, they work by recognizing specific palindromic sequences and breaking the DNA backbone to create either sticky or blunt ends, which can then be glued together with other fragments. Finally, we learned that the cutting frequency of these enzymes is not random. It is mathematically predictable. By understanding the math behind their cutting frequency, researchers can choose the perfect enzyme for their specific experimental needs. You have now mastered the very first step toward understanding how we manipulate the genetic code to create life-saving medicines and genetically modified organisms. Great job!